we'll begin, and I hope that more people will be joining us soon. We're very delighted to have Shlomit Ben-Ami with us today. Shlomit is actually a medical doctor. She did her medical studies at Tel Aviv University. Then she continued to do a postdoc at Tamar Flash's lab at the Weizmann Institute of Science. Tamar does kinematics and motion. And after that, she continued to do another postdoc in Pawan Sinha's lab at MIT, doing it in Project Prakash, which is rehabilitation of vision in rural areas in India. And Shlomit will tell us all about her research. We're very delighted to have you. So welcome to start.
Thank you. I'll tell you a little bit also about myself before I figure out how. So am I still, the screen is shared, right? You see the presentation now?
Yes, we see the screen.
So hi everyone.
Das kann sein, dass das nicht funktioniert.
I'll just mute everybody. Okay, I'll mute the people who are, yes, go on.
Okay. So my name is Shlomit. I recently returned to Israel after a postdoc in the Sinha lab in MIT, and my talk today will be about the biological motion perception after restoration of vision following prolonged congenital visual deprivation. So I also introduced myself a little bit, even though Sharon did. I'm a medical doctor. I studied medicine in Tel Aviv, and then I was a resident in neurorehabilitation medicine in Beit Levenstein with focal brain lesions department under Professor Nachum Soroker, who does a lot of interesting research. And then during my residency, I felt that I really want to understand the mechanism that drive the difficulties in my patients. So in the process of diving into these questions with Soroker, I realized that I love basic science. And then I started my journey in science. My first mentor was Professor Tamar Flash in the Motor Control Lab in Weizmann Institute. I think she might be here as well. And there I studied sensory motor aspects of autism. And I greatly appreciate I learned everything I could learn about science. And then I continued to a second postdoc in the US with Professor Pawan Sinha's Vision Lab. And today I'll tell you about my first project there, working with kids in India that undergo surgery to remove cataracts with the aim of improving their functional vision. So now I'm back in Israel. Our activity in India is on hold, obviously, due to the global crisis. And I'm working on analyzing the data I collected. So I'll first tell you a little bit about the Prakash Project organization and then get to the scientific aspect. Please feel free to stop me with any questions, clarification. I'm happy to have any discussions, and I'm also happy to see more faces if you don't mind putting your video on. And I see a few familiar faces. Hi, Doobie. So this here you can see me with a girl that's running one of the experiments that I'll tell you about. This is a young man who I worked with who underwent surgery this January at the age of 18. You can see that his pupils are completely white and they're obstructing his sight. This is due to cataracts, and they developed probably while he was still in utero, most likely due to a genetic condition. His brother also suffers from the condition, and a few others in his family. This patient's acuity was worse than 20/6,000 when we met him before surgery. And 1 month after, it was still very poor. It was like 20/2,700. But there were high-level visual tasks that he learned, and I want to show you a bit of his and other patients' performance on those. So if he would have been born in the West, then he would get his surgery at age 2 months or something very young. But since he was born into a family in rural India and he and his brother didn't get the opportunity, that's what this project is about. So when Pawan Sinha came to visit his family in India, he became aware of this, and he founded Project Prakash in 2004 with the aim of merging science and service. So these patients are offered treatment, and if they choose, they can participate in studies of their development. And this is an opportunity to test which cues help the development of visual processing in a system that's otherwise already mature. So what we do is we have the outreach, meaning we have optometrists and social workers going out to the field, and they go usually to blind schools. Here you can see some kids standing in line. All are blind, but some of them still have some residual vision. So they test everyone and they see if they can offer any treatment. So it can also be corrective treatment, whatever. We offer all kinds of treatments that could help improve vision, but they look for the ones that have treatable surgeries that can be treated with surgery, and those ones go to Delhi to be tested and to see if it could be helpful to remove the cataract. So helpful meaning that they still have they still have light perception, obviously, and also that they don't have other things like retinal problems that it won't help to move the obstruction. Then those are treated. Here you can see the lens, and here after the lens is removed, and if possible also an artificial lens is inserted. We have very good medical staff, Indian local medical staff, and a very good hospital there. And then the ones who choose to participate in the research, which we'll hear more about. And the fourth part that's new, 2 years ago in 2017 in July, we started up a school. So they learn how to, now some of them know how to read Braille, but they don't know how to read, and they're already older. We don't want them to go back to a blind school. So they can't go also to a normal school. One reason is they're still visually impaired, and the other reason is that they need to catch up. So in this school, and by the way, the headmaster there is herself congenitally blind, and she's a really amazing person. So they start by learning things in Braille if they don't know, but then they move to reading. So for instance, this girl who 2 years ago was operated, now she's reading to me in English. She's doing some math. She's looking very closely, even closer than what you see in the picture, but she's able to do that. These girls learn how to use computers because they're older, and that could help them get a job. Here we're working on categorizing shapes. So we also run experiments in this school, and those are experiments that focus on more long-term things. Here you can see the kids before while they're in the hospital, and we try to engage them in playing ball and drawing if they know some letters. So we keep trying to see how we can progress them, because it's not enough just to improve the sight. They need to get enough input, and we're trying to see which kind of input is more helpful than others through research. So these are the two primary components of the project, the patients and the team. And everyone worked very hard together with a lot of patience to get data, since we collect many experiments in parallel on each child, because we start a small cohort of children every 6 months, and we run many in parallel, many experiments in parallel. So for example, with me, these kids run studies of visuomotor development, other studies testing visual perception of a shape versus the use of visual information of this shape in order to form an accurate and proper grip on it, like you see here, and other tests of how shape processing develops when they get only visual information versus both visual and tactile information. And I think the things that led me to study these specific topics is looking for key features that contribute to learning in these patients, because I think that a better understanding of these learning mechanisms can help us build better plans for rehabilitation, and better maybe apply them in the school, or maybe even immediately after surgery. So in that context, sensitivity to biological motion was a good place to start, because it's a very robust feature of human vision. So this is the first project that I ran. I'm sure most of you are familiar with it, but since this is a student seminar, I want to give a bit of general background before presenting some results. Unless anyone has some questions about the organization of Project Prakash before I go on to the experiment. Okay. So biological motion is simply the motion of vertebrate animals, but it's come to be almost synonymous with these point lights display, or they're also called Johansson figures. These are human figures made of a few dots on major human joints, and as few as 9 dots can be enough to get a compelling percept, but only when the figure is in motion. So here you see a blob, right? You see a few dots. Does anyone want to tell me what you think this is? Does anybody want to guess before I play it? Like what exactly they're seeing here? What?
People are muted, so, but you can unmute yourself in order to answer. I don't know, maybe a person sitting?
Yeah. So it's maybe a person sitting, but when I play it, you see for sure what it is. When the person starts moving, then it's much more apparent what it is, right? So the dots become a very, very strong percept. So what I'm showing you are displays with actual lights, how they used to do it. So this is actually from Johansson from 1970 something. Today, these are made with infrared sensors, and you can control the information. You can take out sensors, you can play around with them, scramble them in space and time, whatever. So this stimulus has really been widely used, and it's really cool, I think, and you can tell a lot about it. For instance, when my husband saw this, he said, wow, this person is really strong. So you can see how strong he is by the way that he's moving. I mean, at least someone that does push-up themselves can recognize that. I didn't. So you can tell, as I said, and I think you saw, that you can tell a lot about the performed action. So performed action, things that people have tried, they did, you can tell what the weight of a lifted box is, what the distance of a thrown object is. You can tell about deception and about intention, which is really cool and amazing. And you can also tell a lot of details about the agent. So what the gender is, the age, emotional attributes, happy, sad, really cool things like personal traits. And you can even, if you know the agents, you can identify individuals. And this information is really useful for social perception. And also, indeed, the brain areas that are sensitive to biological motion highly overlap with those that are active in social tasks. So I think this makes sense generally, but it's also a little bit surprising. And the other thing that they found related to this is that both biological motion and social tasks are impaired in individuals on the autism spectrum. So this is all just to show you that I think this is really cool. I hope you agree. But I really love this kind of stimuli. And it's also other ways to show why, like its robustness is also that it appears really early. And when I say early, I mean early both in phylogenesis. So chicks, pigeons, cats are some of the animals that are sensitive to biological motion. And there's evidence that this doesn't merely result from experience with the stimuli, because even newly hatched chicks with no visual experience show spontaneous preferential looking towards these displays when they're first exposed to them. So similarly, in human development, neonates exhibit preference for the motion of both human and other species when compared with non-biological motion displays. When I say non-biological, I mean, for example, inverted, random displays, rigid displays, all kinds of manipulations. By 6 months, they can already detect directionality of walking, which is one thing that we're going to test. I'm going to show you with Prakash. And the adult performance is reached by 4 or 5 years of age without noise. But when you add noise, and this is a Bachi Khadadze experiment, with you reach much, much later adult performance. So noise really is really difficult for kids, and I think also for many kinds of patients, including it's been shown in amblyopia. So we did not use noise in our manipulations. We used other manipulations, which I'll show you. And another note about biological motion perception is it's really resilient. There's good performance also when stimuli is highly degraded, and also in cases of visual deprivation. So amblyopia, even I saw one study in CVI, and in visual deprivation. So with visual deprivation, even prolonged visual deprivation and congenital. So all the studies that have been done, like 3 or 4 studies, years after congenital visual deprivation, they tested a few things. Like they last VSS, they showed action perception, and there's walking direction, like I showed you before, and there's even a neurophysiological response. We're all normal. We're all at ceiling. We're all comparable to controls, which is pretty amazing. So it seems like specialization with biological motion develops independently, maybe of early visual input by judging by these results of earlier results. So is visual experience required for development of this specialization? Or maybe humans are either born with some predisposition to attend to the stimuli, or maybe you know, our motion, the fact that we move, our system is able to recognize this based on that. You know, many options, And with Project Prakash, we have the opportunity to contribute to this debate. So even though our subjects don't reflect normal development, we can at least ask if the skill is conserved in people with prolonged congenital visual impairment. Is it maybe it's preserved, or maybe it just emerges immediately when acuity improves and enables it, or maybe it's learned. So that was the question. Before I go on to methods, if anyone wants to ask anything. Okay. So first about the methods with the subjects. So we're looking for bilateral cataracts without fundus view. We want a personal history of visual issues or white pupils before 1 year of age. And we're looking for nystagmus. Nystagmus meaning, like you saw in the video before, like the eyes are moving. And this, if the deprivation was early, then nystagmus has to develop. So all our subjects have nystagmus. And here we were looking for visual acuity. We didn't look for very, very poor visual acuity. We just wanted it to be worse than 20/400. That's very poor, but it's not as poor as they used to. Before they would take, Project Prakash would take only ones that had like 0.5 meter. Oh, sorry. My iRobot started acting. And in this case, we just took the definition of legal blindness in India, which is 20/400 or 6/120. And by the way, in the West, it's 20/200. So in India, you need to be more deprivated to be considered blind. Other clues yeah.
Shlomit, can I ask, I mean, you're saying that these are the inclusion criteria. I want to ask if it's an inclusion for the treatment or for the research.
No, so for the treatment, many kids get treatment for many, like even if they have monocular deprivation. And as I said, if we can like help them in the field, if we can give them, visual aids. So this project is screened 43,000, treated about 5,000, and the surgeries we gave to almost 500. So most people don't need surgery. The ones who do need surgery, even if they're not included in the research, if they don't, if they're not suitable for research, they still get the treatment, obviously.
Okay, thanks.
So is this visual acuity after the surgery?
Excuse me?
Is this the visual acuity after the surgery?
No, before.
Before.
So this is the inclusion criteria for research. So we included kids who, in our research, who stood up to all of these, including visual acuity. I'm emphasizing the visual acuity because this is still visual acuity that you can do stuff with. So you can still see, of course, see motion, see patterns from further away. And the reason that I wanted I wanted a variability in visual acuity, because if we're looking at how this depends on visual experience, then we can't only take the very, very worst cases. It depends on the question, but if your question, and so far, every experiment that I did in Project Prakash, they never found any correlation to acuity, but they took very, very poor acuities. So probably under some ability, you don't have a spectrum.
Question?
Yeah.
If you take a healthy individual that with good acuity and you sort of simulate this blur that you have, will they be able to do the biological motion task?
So I tried that. So I took I'll show you what Bengural foils are. So you see here, these are eye tracking glasses without foil. This is with foil. These are not the worst foils. I mean, you can have ones that really, really obstruct. You can see barely anything. And this is just to show you how 20/200 looks. So you can only see the big letter, not anything more than that. And our controls, that's what we do. We give them goggles with foils. And I myself tried with the worst foils, it's LP foil. So it's supposed to be only light perception. But since this is a black background and you have the white luminant dots, and we also enlarge them because of the acuity, then the direction task I could do. That was very easy. I didn't show you it yet, but the walking direction, I'll show it to you next.
But you could recognize that this is a human being, I mean, a biological, you could tell the difference between biological and non-biological.
So I have two tasks. One is just walking and the other is the difference. The difference was harder. So, and I'll just show you control results, which is easier to show. But actually, I don't think that I put down the bottom line. Controls were at ceiling, but we couldn't get to acuity as poor as our subjects with controls. I mean, when we tested acuity with these foils, we didn't use the LP foils on controls, and I think that's what we need to do, because we used the worst kind that like below that, and they were almost all of them very close to ceiling.
Ceiling mean good performance.
Good performance, almost like 90-something performance.
So basically what you're saying is that these patients actually saw, from the point of view of biological motion, they had a perfectly good, not perfectly good, but they had a reasonable visual experience.
Yeah. So, but in life, they don't see these little luminant dots. Yeah. So but for the stimuli, yeah. So even before surgery, I showed them the stimuli. I asked them to tell me what they see, and they said dots or something moving, and I asked them to point, so they could see individual dots. But they couldn't be far. Some of them couldn't be far enough from the computer screen to see all of them together. So they were looking very closely, and they couldn't see the entire percept. But some of them could also do the task. That's what I'm going to show you. Some of them could do the task before. So definitely the stimuli they can see. If they get the experience in real life, that probably depends on viewing conditions and their visual ability.
I want to also mention about the controls, even though you blur their vision, and even if you would reach the same blurred VA or poor VA as your patients, the controls have a very big difference, and that is that they have a lot of priors. So even for the VA table chart, or for the biological motion itself, they have vast visual experience that their system is used to, and that they can try to match to the information to pre-existing templates.
Yeah, definitely. Even visual acuity is an experiment, and since they know what to expect. And I was surprised because I was using foils that were supposed to be very poor, but they were not as poor as they were. So I guess optically they should match this acuity, but it doesn't mean that when you test it, so the person is using cues, obviously. Yeah, it's very interesting. We took a long time to figure out. We did a lot of trials with ourselves and with staff members until we figured out which foil to do. It was a really long process. And I also tried actually blurring on the screen, but what happens with this type of stimuli, if you do a Gaussian blur, then you lose the luminance because it's just white on black. And then I increased the luminance, so I could do that. I can still play around with blurring on the screen, but we finally felt that it's more simulating the situation to use the foils in this case. So I'll show you the experiment. So first of all, walking direction. That's an important attribute of biological motion. And it's by far the most widely used in behavioral experiments, and it plays a major role in assessing if a living creature's intention, for instance, if they're moving towards
Shlomit, your video, I think, is stuck. We can't hear you, or I can't hear you.
Me too.
Hi, sorry, my internet stopped.
Okay, so we can resume to the same place where you were.
Okay, where did you lose me?
It was during the actual presentation of the experiment that you showed.
Okay, I'll go back to that. I just need to figure out why the screen is not working. I need to share my screen again, right?
Yeah.
What do you see now?
The Zoom, we see your
Okay, wait.
browser. Yeah, that was the
That was the presentation.
yeah, this is the browser that we're seeing. And yeah, that's the presentation. Yeah, I think that's where we were.
Okay. Oh, wait, just a second. Sorry. I have to share something else. Okay. Okay.
Yeah.
So I don't know, I should show the experiment. I don't know if you heard the background part. I want everyone to guess, like, if I can see your hands or something, if it's right or left. This is going to be fun because it's easy. Wait, I can't control it for some reason though. Here. So right or left, you already have the answer. So as you can see, this is a really boost for self-confidence because it's a very, very easy task, and the patients get to see it for as long as they want. And I was saying before that this is the most widely used in behavioral experiments, and you only assign a walking direction to something if you see it as animate. So one of our collaborators, Nico Troja, who collaborated on this project, so we took all this data from him. He had one experiment that he found that rating of animacy attributed to walker stimuli are linearly correlated with the ability to discriminate the stimuli's direction of walking. So there's a reason, I guess, they use this kind of stimuli very widely. Oh, can't control the screen. I'm trying to go, oh, here. So in this experiment, we asked, as I said, we first asked them to point to individual dots. And then they just had 16 trials that had to say right or left, like that, like you said. And correct response is right or left, if they're correct. They could point, they didn't have to say right or left, but at some point they already learned the right and left in English, and they would say it to me. So the other experiment we had is this sort of thing. This is the most widely used imaging task, is showing this coherent version of an action and then manipulating the same actions. I will show you. So there are 2 second clips of actions, which we showed I'll show you how the experiment looks, and a manipulated version of them. You see, these are the same exact movies, but we scramble in space the location of the dots, where every dot begins. And in imaging, that's what they use to locate the posterior superior temporal sulcus area, which is the hallmark of biological motion perception. And it's identified by this contrast. And these subjects, the reason that I showed them these 2 second clips is that they were also in the magnet. Most of them, about 2/3 of them, we were able to get in the magnet and see this stimuli. But in the behavioral experiment, they didn't only see that contrast. They also saw 2 other contrasts, which is inversion and phase scrambling. And they saw 8 different actions, but each was performed by 3 actors. So it was 3 repetitions, but it was taken from different actors. So it's not an exact repetition. And they were asked to identify, to point to the person. And if they identified the current action as a person, then we consider that correct. So this is what the experiment looked like. So we said, point to the person, and we were very careful to make sure that they're looking, because it's only 2 seconds and it's hard to focus them. So we would say ready, and then start the experiment, and then say, like, point here, is this the person? Is this the person? Where was the person? And here you saw scrambled. Here you're seeing a person sitting down, and this is, I think, phase scrambling of the same one. So phase scrambling meaning that each dot starts now in a different time. And finally, I'll show you inversion, I think, here. So a person sitting down, and then the inverted display of it. And usually what, just to tell you about before I go to results, if there's any questions about the methods. So like these 3 manipulations, I was expecting that maybe, since it's considered, I mean, from other experiments, that inversion should be the easiest, then the spatial scrambling and temporal scrambling should be the hardest. So I was expecting that maybe we'll see these differences and we'll see how they develop independently or not independently, but some didn't have any difference between the 3 types of manipulations, and the ones who did have differences, I didn't see any trends. So I'm going to show you collapse results of the 3 types of manipulations. So some results. First question, is beam perception preserved despite the low vision? So the answer is it depends. So in some subjects, it was preserved. So they could do both tasks before surgery, above chance. So when I say above chance, I just mean that a statistical test determined that. The controls were a little better, but they were still above chance, these patients. And then there were 9 patients who could not perform it, who were below chance before the surgery. And we also had 2 subjects that were mixed, so they couldn't do the actions, but they could do the walking. And as you saw, the actions is a much harder task for various reasons. So these differences, might they be driven by acuity? So this is a very close to not being significant, but still a significant difference between the 2 groups, the chance and the above chance group. But more interestingly, there's a correlation between the acuity and the performance. And you see here different time points. So this color is pre-operation. These are all the subjects and their performance. This is for the walking direction, and this is for the coherence, discrimination, and actions. And then after the first operation, so I'll show you soon that they improve. Most of them at least improve on the task, but the correlation persists until it persists after the first and the second operation. And they get to almost ceiling performance, depends on which task. They get earlier to the ceiling performance in walking direction. And then I stopped showing already here the 4 to 6 month because they're already all of them here at ceiling. And the correlation persists longer in the actions. For normally cited age-matched controls who are almost matched for acuity, we didn't see such a correlation. We want to continue and collect a few more with higher blur. So this is, I'm sorry, I'm just going to close the door because there's a lot of noise. Okay, sorry. So here we're sort. I mean, I think this is the first interesting point, that it depends on your acuity, if you're able to do the task and how much you improve. So does acuity improve after surgery? So the answer is generally yes. Most subjects improved and are like beneath this diagonal line. Sorry, I just want to say that this is the acuity prior to operation, and this is the acuity 1 to 2 months post-operation, and we continue to also test them afterwards. And the catching up of biological motion perception. So with the walking direction, which is the easier one, we said we had 2 groups. One was the chance pre-op performance, the other was the above chance, which continued to be above chance, and the chance performance, they all caught up. And they did it within, I don't have this here, but this is pre-op, post-op, post-first operation, post-second operation. This is 1 to 2 months later. This is 4 to 6 months later, and this is 9 to 12 months later, which we don't have still for all the subjects, but they caught up before that. So around 6 months, they already all were caught up. For the action coherency, it's not as clear-cut. So this is the above chance group, and the below chance group, they improved but didn't quite catch up. But then if you look closely, you see that there are some subjects that did catch up, and others that were really stayed exactly the same performance. So I'm showing you here just the division, only the group that was chance pre-op here. So I divided them into purple and orange. So all of them caught up, as we saw in walking direction discrimination. The mixed ones were already above chance. The mixed ones are in green. And then for action coherency discrimination, we see that chance pre-op caught up. These are 6 subjects. And then there are 4 that just didn't catch up. They just didn't change anything in their performance. And the mixed ones also did not improve. So we have 4 purple ones and 2 green ones, meaning we have 6 subjects that didn't catch up at all with action coherency discrimination. And there are several options for why this happened. So can acuity explain this difference? So in this case, there are not many subjects, but we don't see a difference in acuity. Not an improvement in acuity, and not an acuity pre-op or in follow-up. So we're looking at other data to see why this, to try to see how to explain this or what this relates to. So the first thing to mention, as I mentioned before, this is a harder task. It's only 2 seconds. There is a matter of attention and of memory. And the reason that these tasks are so different because the second task was actually supposed, goes along with the fMRI experiment. But we do have the fMRI data. So we have it for representatives of each of the 3 groups, the ones that were chance before, the ones that were above chance before, and the ones that improved and didn't improve. So I still am pre-processing this data, so I don't have even a hint to give you, but I will in a few months. We also, when we tested these people and we asked them to say which was the person, there was a follow-up question, which was, what was the person doing? So they gave us action descriptions, which we took and we're analyzing now. And we also, when they were describing, we mentioned if they imitated. So we can also see ones who imitated more or less, because many people, when they say, what was the person doing? They say, they do some, like they answer, but they also do some movement. And then with imaging, we have this localizer of the coherent versus scrambled motion. And we also have resting state. So we can look at the functional connectivity during rest, which is going to be very informative. And of course, also at the functional connectivity during the task. So all of this is going to come.
Shlomit, you don't remember if you could cluster them or split them according to their successful imitation? You haven't looked at these data yet?
No,
whether the ones that improved have successfully imitated the motion.
Since because of the way that this data is saved in very different ways, some of them is handwritten, most of it is in Excel, so it needs to be organized. So I can't even tell you, but just because it's a very messy process to run different people running different things in India. So unfortunately, I need to still organize and look at it. But I can tell you from when I ran the experiments, most of them didn't imitate. So yeah, there were a few that imitated more, but I don't exactly remember who, but I remember there was not a lot of imitation at the beginning.
I have another question, whether you can still have the researchers rate their, their fluency or the way they move.
Whether you mean in general, their general movement?
In general, yeah, in general, if you could do some rating of how their movements are actually perceived by normal observers. Maybe.
Yeah, I have more than that. I have recordings of their movements. So since I let them do some simple things like reach to grasp, and then it will be not only rating, it will be recordings, it will be, even kinematics, I hope I could get from that video. Yeah, of course, it's a very, very interesting question. But again, you need to be patient. So there's data.
We'll invite you next year for a follow-up, of course.
Yes, thank you. At least I have time now to do all of that because I'm not running new experiments or follow-up experiments. That's the only good thing about this hiatus. So just the last piece of maybe a clue about how experience is related to this to this task is the ones who catch up. Does it happen gradually, or is it like when they get the improved acuity, which is immediately after surgery, do they just immediately jump? And I have to say that some of them I tested like 1 day after surgery, and others, there's a week to test. Others I may have tested 5 days after surgery. So like in hindsight, I should have done all of them on the same day, but it's not really possible. So here what you see is, you see all of the subjects that were chance pre-op in the walking direction. You see that we're missing some time points for some of them, but that's how they were pre-op. And then you go like until you get to the 1 year, 9 to 12 month follow-up. And some of them improved gradually, and some of them caught up even, there was one who caught up almost immediately. But there are some that improved very gradually, and only reach it at the second follow-up, like 4 to 6 months later. This is the easier task. With the harder tasks, there are the ones that don't improve, and then the others are, I don't think anyone immediately reached ceiling. Most of them didn't even reach ceiling at all. So some of them at least have a gradual improvement. These are the ones that could perform at above chance. Here you see the ones that we tested this January, and we can't keep testing them. They were actually the worst acuity ones in this group. And with action coherence, even the ones that were above chance but were not at ceiling. So I think you could say generally that half at least have this gradual improvement. So another clue. So to summarize, so the ability to retrieve walking direction, to discriminate coherent actions from point light displays is correlated with the visual acuity of the subjects. And it's not preserved in all subjects, as maybe we could have thought from previous results. So if you have bad acuity below a certain threshold, then you can't do this task before surgery, even though you can see the individual dots. You may not be able to see the whole percept, but also right after, you don't improve immediately usually, especially the ones with the poor acuity. And two clues that suggest dependence on visual experience. One is the correlation between the performance and the visual acuity in patients, but not in control so far, and the gradual development. So some follow-up questions, and I'm showing these specific questions because I already have experiments about these ones. So we saw learning. So which type of information contributes? Both to the learning, like in the learning phase, which cues are important? And when you already master the task, when you're already at certain performance, which cues are you using? It might be separate things. So we're especially interested in the ones that are important for the learning process, but these could be useful for us. And you might become independent of those later. And the other is not only which cues, but also which behavior contribute to acquiring these skills. For instance, looking behavior, which we collected, for instance, for the actions. We don't know how useful it will be, but since the actions are very separated between each other, then you could use, even though their eyes are moving like this, you could see which one they're looking at more. So maybe even the ones that didn't improve, they might be looking differently at each of these, at each of the, like at the manipulated and unmanipulated. So this could be a hint. And also for the walking, we're already going into these questions and already have results about these two questions. You'll have to wait. These I can talk about whenever you want. Okay, so I just want to thank the Prakash patients and the families, which are really amazing. The ones who come and take this brave step of doing the surgery at this age are amazing, amazing people. My mentors over in science. We have a wonderful outreach team that goes out and find these people, and then they also are with them when they're in the hospital. They're the go-to people. The medical team, the amazing doctors and surgeons. This is our research team in the past and the present. And this is the new education team that I was telling you about, also very amazing. And there were people who helped me with technical issues, the collaborator Nico, who provided all of the stimuli, and Ella, who I'm working with partly on analyzing some of the fMRI data, and students. Thank you. Questions?
Thank you so much, Shlomit. Let's all give a nice applause. It was a wonderful talk. And everybody can unmute themselves and ask questions. I'm sure people have a lot of questions. This is a fascinating project and fascinating results. So you're most welcome to ask questions, of course.
I'll try to unshare the screen.
Can I ask a question or two?
Yeah.
First of all, thank you, Shlomit, for a wonderful talk. And I think kol hakavod on this project. I think it does a lot of wonderful things for people. A question, it's actually a follow-up question of what Sharon was asking about the motor abilities. First, I was wondering if any of your patients or the people in the research have motor disabilities, and do they, so meaning they're paralyzed or in wheelchairs or have actually no walking experience, and does that affect their percept? And
That's a great question.
and then my second question, which is associated, if you after post or pre, have them practice the actual actions that they're supposed to see later. So sort of like an embodied learning. If they actually do actually perform the motor action, do they improve in their percept?
So this is exactly the kind of question that I would like to study. We haven't done it, but since we have them in the school, that's what I was thinking, to have some physical exercise kind of class and have them maybe experience some of them, because they really don't have any experience with balls. I mean, of course, they sit down, they get up, but some of these things, and we can find more that they don't have experience with, and having them do some of them. So first of all, yeah, definitely I want to do that, but not yet. It's pretty hard also to control, to measure. And so that's one of the good things about the school environment. The first one about disability, so we actually don't have people, since we're trying to look at a brain that's as normal as possible, except for the visual issue. We exclude anyone that has other brain dysfunctions, or generally other dysfunctions like mental or physical. So we don't study these. I mean, if we actually didn't also encounter many. I mean, it's a subject by subject decision. So if we find anyone that's only physical, especially if it's not brain related, then we definitely might study them. But we decide based on each patient, because we only have, it depends on the cohort. We can only work with up to 10 subjects. Like my first trip, we worked with 10 subjects. It was really, really hard for them. There's not a lot of space. One problem in India that we really don't have the space.
Do you collaborate with other projects like this in the world?
No, we're a really small endeavor, so we don't have a lot of resources. I mean, just time and people. Mm. But it is something that we'd like to do, definitely.
Thank you.
Can I ask? Oh,
okay. Go ahead, Rafi.
Do they get feedback about the choices during the testing? In other words, can they use the testing to train themselves to improve?
I guess they probably are training somewhat from the testing, but they don't get any. I make sure that they never get any, even like nada, or like, which people sometimes do when they don't notice, but everyone is trained not to do that.
Okay.
I saw that you have eye tracking glasses. Do you have eye tracking data? What do they pursue, follow the biological motion or stuff like that? Do you have?
I have eye tracking data from the glasses and also from a screen-based Tobii. Only at Followup 2 are we really able to calibrate. Before that, it's partly calibrated. Still, so I don't know how much to believe it, but still we have the data, and we also have like how reliable it is. So we could exclude the ones that are not reliable. But it's with the glasses, it's a little hard because you really need to start coding it individually, right? Because you can't do it automatically unless you use computer vision or something like that. So I tend to use this data just to see the ones, maybe like just to compare the ones, for instance, who didn't improve, to see if they were maybe didn't report, but they were still seeing the difference, like one use of this. But you can really use this only if you plan the stimuli accordingly. So it can be useful for eye tracking. For instance, what I did with a screen is that I took a stimuli of a person walking, and I wanted to see if they were looking at local cues. So I spatially scrambled everything, but only not vertically, only horizontally. So still the feet are under the head, and everything is like the correct order. And then you could just look if they're looking at the feet or at the center, or at the head. And in this case, the scrambling is done so that you can only get local cues, because you don't have any spatial information and you barely have phase information, but you do have like the 2 feet who look like they're in a wheel. And then you behaviorally also what you do to the stimuli is you take each of these individual dots and you also manipulate it so that you take the average location and you switch it. I forget the word. So now behaviorally I can see that if they're correct when it's going the right way and it's incorrect when it's going the opposite way, then I know behaviorally they're relying on the feet. So I have the behavioral result. Now I want to know if they're looking at the feet. So I do this while they're having, and I just want to see if they're looking at the center, the top, or the bottom. And then I also invert that. And then I have exactly the same AOIs, but it's a different stimuli. So that's like the way I found to go around the problem that they have this nystagmus, and it's really hard to follow. But again, this I can only do at follow-up. I can compare follow-up 2 to 3, like 6 months to 9 or 12 months. I can do it pre-op, I just can't calibrate.
But it's, I mean, you can get some information without calibration. You can get
definitely
information about the type of eye movement, depending if you are able to track it. If
no, definitely. So that's why we were using the glasses.
Get any data, there's nothing to do. But if you do you can really get interesting information without any calibration. Something.
So that's why we were using, when we couldn't get calibration, we were using the glasses. The reason we were using the glasses is because there you can record the samples, even if there's no calibration, and even if it can't have any fixations, we still have the samples. So yeah, we have data for that with most of these.
Okay, I'll be glad to talk with you about it.
Yeah, I know, we will. Anyone else?
So one more question. You mentioned nystagmus. Do they continue with the nystagmus after the eyes are open? And is there some kind of reduction in nystagmus over time?
Yeah, they continue for the entire life, but it seems like there is a reduction. We're still, so right now we're taking videos, we're measuring that. It seems like there's a reduction over time, and also when they're concentrating, it sometimes gets worse, but then so you can do some behavioral things to help it reduce. And we're just like trying that out, not experimentally, just to see. So we're still exploring that. But yeah, nystagmus is a very big issue. And I think if you can address
I wonder, do they feel the nystagmus? In other words, the world jumps to them, or it's completely compensated perceptually? They see the world spinning or something of that sort, or that might explain.
It's a good question. I think from what I read, because I don't have the Hindi to ask them, it's a pretty complicated also to explain. We could try though. But I think it impairs their acuity. It's one of the things that impairs their acuity, on top of the fact that they didn't have that they didn't develop in time. But it definitely impairs their acuity. I mean, my sense is that they don't see the world jumping, but of course I can tell you that things like shape perception, it's really hard for them to see complex shapes. So what's the reason? Is it because the spatial reference frames, or is it because these jumps? So that's why we try to let them do things like match the shapes, draw the shapes, discriminate the shapes, many things to see the differences and try to figure out where this comes from. But I don't have an answer.
So
can I'm sorry?
Go ahead.
Okay, are you letting me ask?
Yes.
Hi, Shlomit. Hi. I just wanted to know if you are, when you're collecting data from the copying or drawing, or do you have like a detailed data? Have you looked at the kinematics of the trajectories?
Unfortunately for that, so far there's just a video, but you could extract the XY samples. It's something that can be done, but it's not. It wasn't collected in a way that is very reliable, this data.
So you cannot really extract from these trajectories or something, no?
Also, it's not repetitive, so it's like they do one because it's really hard for them. We could maybe get some, but the experiment was not tailored to that. It's really not easy for them at first to do it, especially on an iPad and stuff like that. But definitely one of the things that we could improve in our.
Because you say you have they have difficulties perceiving the shape, right?
Yeah.
So have you looked at some kind of gross comparison to the shape they reproduce, or
Yeah,
some kind
that
of
we have.
assessment?
That we
So what
like
kind
the,
of, what measures do you extract, or what kind of features you're looking at?
so it's just geometric features. This is actually not my experiment, but it's actually geometric features of the template versus, so there's a template, like you do it on the template or you do it from memory. The similarity, if you're crossing over the lines when you're doing a tracing movement. But even just by looking, you see that at first their squares are really circle-like, and it takes time until they make the edges. They also do it from tactile. They feel it and then they do it. So there are a few, it's very, very initial data. What we see is we see some development in the shape. And that also depends if the shape is complex or primary. It depends on, like, there's these overlapping shapes also. Like, so that's, or touching shapes. So it's more, right now it's more a matter of geometry and less a matter of kinematics in this point.
But Shlomit, you can actually join, I mean, not measure the kinematics, but with respect to what Tamar has asked, you can actually show these videos to naive observers so that they can give some judgments about the smoothness of the motion and the precision of the action, or so you could get these ratings from people who are naive to the situation.
Definitely, I can tell you that all of them are not smooth. So definitely the motion is very, very, very jumpy. They keep checking back and forth. They don't look at something and then they remember it, and so they use the stimuli while they're, and also from memory, it's not smooth. They make a lot of mistakes, even though they know that they're making the mistakes. So they realize that they're not doing, but they just can't do it better. Some of them, they can say, no, this is not working out for me. Smooth, it's not. You can rate maybe how smooth it is.
Yeah, precisely. Maybe there's a gradual change into becoming a bit smoother.
Yeah.
I don't know.
I mean, if I wanted to see the smoothness, I would give them repetitive actions. I think that, like, what we have is probably not enough to see the smoothness just per se, just because they're not practicing it. They're not even close to getting some skill developed. So, but that's something we could do, like give them repetitive
actions.
Drawing. Yeah, drawing or just natural actions.
Okay, so let's thank Shlomit again for a wonderful and exciting talk. And I hope we'll be able to host you next year as well.
Yeah, if you want me to continue to talk about the continuous questions that I already have ready, or other subjects that are
great. Okay, so we'll be in touch about that, but not here. I mean, outside the Zoom. Lovely. Thank you very much.
Okay, thanks. Thank you, everyone.
Thank you. Bye-bye.
Thank you.